Hello, everyone. Welcome to my spatial data analysis video tutorials. Today, I'm going to show you how to analyze the Nanostring mouse brain dataset. So you can download the data from the link I showed here. We are going to use the coronal hippocampus and the cortex dataset. If you want to just create a short object, you can download all the CSV files from this link. So I downloaded the data already. You can see in my folder here for the quarter mouse brain, we have the expression matrix data, we have the field of view position files, and we have the metadata because we are going to create a short object. So we are not going to use the metadata here. Then we have the tx file. This is the file to save all the molecule positions on the image. Then we have the polygons file. We can go to R and use the data here to create a short object. Let's go to R. You can see I have the code written here already. So we need to use short and the tidyverse package. You can create the short object from CSV files, or you can create a short object from the TileDB data. Also, when you export the data from the Nanostring CosMx shape analysis, you can also export a short object. So today, I showed you I downloaded the CSV files. We are going to create a short object from the CSV files. So we can use the load nanostring function from the short package. We tell R where we saved the data. Then we can name the field of view as the brain. Then C will be nanostring. If you have a different project, you can give a different name for the field of view. So if we run this code, we will generate a short object. Let's run. You can see R is loading the data. Okay, you can see we created a short object. We named it as the nano object. We can have a look at the object. Let's connect the short object. You can see now in the object we have the C list 1. We only have the C names as nanostring. We have the layers to save the original count. And down here you can see we have the image data. If we connect the images, you can see we named the field of view as brain. We connect the brain. You can see we have the molecules. So the molecules data slot save the molecules position on the image. This data comes from the tx CSV file. And also we have the boundaries. You can see in the data slot boundaries, we have the centroid and we also have the segmentation. So the centroid and the segmentation data comes from the field of view and also the polygon files. So at the moment you can see the reduction is 0, command is 0 because we haven't do the analysis yet. So now we can have a look at the row names because this data set only uses the 950 genes. If we have a look at the row names, you will see we have all the gene names here, and also you can see from 951 to 960, we have 10 negative probes. So now we can analyze the expression matrix data to cluster the cells and generate the UMAP. Normally, if we run the standard workflow for short cell clustering, we can run data normalization, scale the data, and find the variable features. You also have a choice to use the SC transform method. The recommended method for spatial data analysis is the SC transform. If we run SC transform, we will normalize the data, scale the data, and find the variable features. So today we are going to use the SC transform method. Let's run. So if we run SC transform, we will normalize the data, scale the data, and also find the variable features. Because in this dataset, I showed you, we only have 950 genes. So we will use all the gene names as variable features. So we finish the SC transform analysis. If we click the thread object, you can see now in the cis node, we have SCT data slot. So that's the analysis performed by the SC transform. You can see in the command we have a command list. We run the SC transform analysis. So after SC transform, we can run PCA. After PCA, we can run UMAP. Now we can find the neighbors, and also we run find the clusters. I'm using the resolution 0.1. If we use low resolution, we will get small numbers of cell clusters. You can see we got 16 cell clusters at resolution 0.1. So now we are ready to look at the cell clusters. We can use the dim plot to look at the cell clusters. Okay, you can see in my plot window, we have the cell clusters. You can see we have 16 cell clusters from cluster 0 to cluster 15. So we got the cell clusters. Now we can look at the cell clusters on the image. We can use the image dim plot function. We need to set the field of view as brain. We named the field of view in the beginning as brain. We can use the color as polychrome to generate the image. Let's run. Now you can see, we can see 16 cells clusters on the image. You can see, for example, cluster 6, they are the cells in the hippocampus. And you can see cluster 8, they are the cells in the ventricles. So the cell clustering worked very well. You can see distinct location for different cell clusters in the mouse brain. So we use the color as polychrome. You have a choice to use a different color. For example, you can use the color as Gatsby. Let's run this code again. You can see we generated the image again, but now the color coding for each cell cluster are different as the polychrome. So we got the cell clusters, and also we can see the cell clusters on the image. Now we can annotate the cell clusters. You can use single cell RNA sequencing data analysis as a reference dataset to annotate your cell clusters. If you have a dataset from the normal mouse brain, you annotate the cell clusters using reference data will be okay. But if you have a dataset from different disease or different treatment, then the gene expression profile will be changed. So it is very difficult to annotate the cell clusters using the reference dataset. So here I'm going to show you the traditional method, use mark genes to annotate the cell clusters. Normally you should know well the mark genes for the cell types in your tissue. If you are new to this field, you can do some reading for previous single-cell RNA sequencing data analysis. They will indicate the best mark genes for different cell types for your tissue. I'm working on the lung tissue and the peripheral nervous system. I'm not expert for the central nervous system, so I'm going to use some mark genes to demonstrate the method for cell canasta annotation, but I'm not going to the details for the subtypes of the cells, for example, the neurons. So we know the PLP1, MBP, and the Wernicke-1 TFAP are mark genes to enable glial cells in the brain. Then we can use those mark genes to identify the glial cells. Now we can use the feature plot to look at the mark genes expression in the cell canisters. We can run the feature plot to see the expression for those genes. Let's run. You can see PLP1, MBP, they are myelinating genes. They have high expression in canasta 0. Then Wernicke-1 labeled oligodendrocyte and also oligodendrocyte precursor cells. So canasta 0 and 11 express high-level Wernicke-1. Then TFAP is expressed in canasta 3. So cells in the canasta 3 are astrocyte. So next we can use some mark genes for neurons. For example, the CCK is a neuropeptide, and the SLC17A7 is a mark gene for neurons. Let's run the feature plot for the gene expression in neurons. We can run. So you can see from the feature plot, for example, the KLM2, they express high level in the canasta 6, 7, 10, 12, 1, 5, 14, 4, and 13. So we consider those cells are neurons. Next, we can run mark genes expressed in the cells associated with blood vessels. For example, the CLDN5, PCAM1, they enable endothelial cells, and the RGS5 enable parasite. So if we run, so from the feature plot, you can see we know canasta 2 are the cells associated with blood vessels. So also in the brain, we have the microglia. You know microglia, they express similar genes as the immune cells. Then we can use the immune cell mark genes, for example, C1, KOA, C, CTSS, to identify the microglia. Let's generate the feature plot. So you can see those genes are expressed in canasta 9. So we know cells in canasta 9 are microglia. So this is the method we normally use to annotate the cell canisters for single-cell RNA sequencing data analysis. You can see it still works here. But for spatial data analysis, we also have an advantage to look at the cell canisters on the image. It helps us to name the cell canisters correctly. For example, we can use the image dim plot function to look at the localization for each cell canisters. If we look at the cell canister 6, let's see the cell canister 6 localization on the image. If we run, you can see we generated the image. Cells in canister 6 are in the hippocampus. So we know they are neurons. If we look at the localization for canasta 4, let's run. From the feature plot, we know canasta 4 are neurons. Then you can see here, they are the neurons in the midbrain. From the feature plot, I didn't show you the mark genes for canasta 8 and 15. Then we can have a look at where are the cells for canasta 8 and 15. Let's run canasta 8 first. You can see cells in the canasta 8, they are in the ventricles. Then canasta 15 are the cells inside the ventricles. So now from the mark gene expression and also the localization of each cell canisters on the image, then we can name the cell canisters. So if you don't know well about the mark genes, another way you can use is to find the mark genes for each cell canister. For example, we can identify the mark genes for the canister 6, use the find the mark genes function. Let's run. You can see we have a data frame here named as marker 6. If we click the data, if we arrange the log2 folder changes from the largest to smallest, you can see cells in cluster 6 may express high-level PPFA2 summer 5A. This is the action guidance molecules. Also, it expresses like a BDNF MPY1 receptor, so we know those genes are expressed in neurons. This is another way you can identify mark genes for each cell cluster. So now we can use the feature plot to look at the two highest expressed genes in cluster 6. If we run, you can see both genes are highly expressed in cluster 6. We can also look at the gene expression on the image. We can look at the PPFIA2 first. You can see from the red color, this gene is highly expressed in the neurons, and we can do one more demonstration for the stem 5A. Again, you can see high expression of stem 5A in the neurons. So I showed you how to annotate the cell clusters, use the mark gene expression, and also based on the localization of different cell clusters. Now we can simply name the cell clusters. I showed you cells in cluster 2 are cells associated with blood vessels. We can simply name it as blood vessels. Then 0 is oligodendrocyte, 11 is oligodendrocyte precursors, then 3 is astrocyte, and 9 is microglia. Then cells in cluster 8 and 15 are the cells in the ventricles. Then other cells are neurons. So now we can name the cell clusters. We can use the rename ident function. If we use the dim plot to look at the cell cluster now, let's generate the UMAP. You can see now we named the cell clusters neurons, OPC, oligodendrocyte, blood vessels, microglia, astrocyte, and cells in the ventricles. So I use the simple way to demonstrate the method for annotated cells. If you want to name the cell clusters for your dataset, you probably need to go more details. For example, the neurons in different cortex layer in different region of the brain. For example, the cells associated with the blood vessels, we have endothelial cells, smooth muscle cells, and parasites. So you can use the method I showed you here to annotate your cell clusters. So we named the cell clusters. Now we can look at the cell clusters on the image again. You can see now we show the cell clusters by their name on the image. You can see now all the neurons are in green color. You can see all the cells in the hippocampus, C1, C3, and the dental gyrus region, they are in green color. Then cells in the ventricles, they are in yellow color. Oligodendrocytes are in red color. So we cluster the cells and also annotated the cell clusters. Now we can look at the gene expression on the image. So we can use the image dim plot. We can use olig-1 as the example to see the olig-1 expression on the image. You can see, use the image dim plot. We label the olig-1 expression cells in red color. At the same time, we show all the cell clusters. Next, we can use the SLC17A7. It is the mark gene for neurons. We can generate the plot. Okay, you can see now the SLC17A7 are in red color, and also we have the cell canisters here. If we put both genes together, we can look at the co-expression, use the same function, image dim plot. If we run, you can see now on the image, we have the oligo 1 in red color, SLC17A7 in blue color. Because the oligo 1 is expressed in oligodendrocyte, then SLC17A7 is expressed in the neurons, then they don't show co-expression. If you don't want to show the cell canisters, you can use the image feature plot. For example, if we run the image feature plot function for the gene oligo1 and the SLC17 and A7, if we run, you can see now we generated a separate feature plot, like the feature plot on the cell canisters. We show only one expression. They are in this area in red color in oligodendrocyte, then SLC17A7 expression in neurons. So I use some genes to demonstrate the feature plot, image feature plot, image dim plot function. You can try other genes that you are interested. So now I'm going to show you how to zoom in an interested area to look at the gene expression. So the thread function has a crop function. We can zoom in to a specific area. First, we can look at the image again. Now we use the axis as a tool to label the x and the y axis. Let's generate the plot. You can see now we generated the feature plot. We have the x-axis and the y-axis. So for the demonstration, I'm going to zoom in this dental gyrus region. If you look at the x and y labeling, this area we can set as minus 2500 to 100 for the x-axis, then y. If we select 145 to 150,000 area, we will crop this area from the full image. So let's set the future global maximum size. It will depend on your computer. If you have a large memory computer, you don't have to set it. So now we can crop the region we are interested in. We name it as the hippocampus crop. Okay, you can see we generate a new field of view object, hippocampus crop. We want to see the gene expression in this region, so we need to give this field of view area back to the nanostring object. Then we can look at the gene expression because we have the gene expression data in the C data slot. By default, we are looking at cells from the centroid position. Because now we can zoom in, we can set the default boundary as segmentation, then we can see individual cells. Let's set the default boundary as segmentation. Now we can use the dim plot function to look at the zoom 1 area because we cropped the dental gyrus region and name it as zoom 1 in the thread object. So we can run the image dim plot function for the zoom 1 area. So you can see now we focus on this area, we zoomed in. You can see the morphology for the dental gyrus. Now you can see individual cells because we set the default boundary as segmentation. So the data was connected by individual hybridization dot from each cells. So now we can actually look at the molecules. For example, we look at both omega-1 and SLC17A7 molecules position on this image. Again, we can use the image dim plot function. Let's run. You can see now, you can see the dot. You can see omega-1 is in red color near in oligodendrocyte. Then SLC17A7 is in blue color. You can see individual molecules in the cells. So that's the demonstration for this dataset. Now we can save the data we analyzed for the quarter mouse brain dataset. Next time, if you want to look at other gene expression, you can just read the data back into R. You don't need to do analysis anymore. So I'm going to stop from here for today's demonstration. I hope this video can help your Nanostring spatial data set analysis. So if you like my video tutorial, please share it with your friends. I hope to see you in my next video tutorial.